I am Marc Mechelin, CSO and one of the 3 co-founders of CleanCells in July 2000 in the west coast of France. At the very beginning, we proposed the detection and eradication of one single contaminant, the Mycoplasma. Along the years, and in response to the request of our clients, we have developed a large panel of tests related to the safety of bioproducts. Through our services, we aim to accompany our clients and partners from R&D stage up to late clinical phases and market launch.
I am François Pedlaborde, Business Development Director at CleanCells. CleanCells is a GMP-certified CRO and CMO involved in the quality control of biologics, including vaccine, protein, and cell and gene therapy products. But also, as one-stop-shop solution, we propose cGMP manufacturing of starting materials and associated quality controls. Our goal is to propose a comprehensive panel of testing solutions in accordance with worldwide regulatory requirements to support the development of our customers' biologics. We perform over than 7,000 analyses per year in order to ensure safety, identity, and purity of biologics for our customer base all around the world. Our agility, quality of services, and scientific expertise are the key value for CleanCells to ensure and to guarantee the success of our partners' programs.
I'm Lucie Tulot, the head of manufacturing department at CleanCells. We have the capability to manufacture mammalian or human cell line in static phase or in agitation using different types of technologies and various banks according to the GMP guideline. We have 8 manufacturing units with positive pressure and 1 unit with negative pressure, all qualified in place.
I'm Gwenaël Autrette, Head of Quality Control. We have dedicated laboratory areas for our different activities. Microbiology, where we carry out sterility tests, Mycoplasma and Mycobacteria detection by direct culture, cell biology, especially for in vitro detection of viruses, and molecular biology for the specific detection of contaminants such as viruses or Mycoplasma by PCR. We also have capacities for karyotyping, a biosafety level 3 laboratory, and a dedicated sequencing laboratory with capacities for NGS.